Okay, so we've seen these before. This is the only part of the first part of chapter 11 slides that made it. It made the cut, apparently. And so real quick, we need to finish up talking about the odds ratio and relative risk. But before we get directly into those, we need to remind ourselves of the difference between a retrospective and a prospective study. Okay? Now, both of these tend to be hypothesis-free analyses. Both of these tend to be hypothesis-free analyses. However, some of our prospective studies on occasion are hypothesis-driven, but a lot of these typically are hypothesis-free studies. And so here's the way a retrospective study works. We take a group that has the pathology associated with a particular genetic illness, and we hope that our diagnosis is right. For all intents and purposes, they have the genetic illness that we are studying. That genetic illness might be Alzheimer's, it might be type 2 diabetes, it might be a specific type of cardiovascular disease, whatever it is. So we've got our, what we call our experimental group, and they have that genetic illness. Then we need a control group, and these are people that do not have that genetic illness, at least at this point, right? And you have to remind yourself, this is challenging because many of these complex genetic diseases have a late onset, right? Many of these complex genetic illnesses do not manifest themselves until late in life. So there's a challenge. On top of that, so that's a challenge with your control group, is some of those individuals, they may at some point in their life develop that genetic illness. On top of that, they're hard to find because people without Alzheimer's rarely go to physicians that specialize in Alzheimer's, right? That makes sense? If you don't have Alzheimer's, you have no business interacting with a physician that specializes in Alzheimer's, the physician that is going to be doing these retrospective studies. So they're really challenging to find, meaning you need to recruit people for your control group. And whenever you're recruiting people, you're always worried about sampling bias. Okay? When you're recruiting people for clinical trials, you're always worried about sampling bias. You know why? Because the people that volunteer to do medical trials when they don't have the genetic illness usually are weird, right? They're just strange people. Or they need something, right? And so it's like, maybe we're dealing with another genetic illness there, right? The need to just be part of every clinical trial you can be involved in. And you're like, well, maybe they need money. The money's really not that great. It's really just, I don't know what it is. Like people that need money, you donate plasma, right? You don't need to be part of some medical trial. So they're hard to find, subject to a lot of sampling bias. However, I mean, sometimes that's what you have to do. Okay? Now, a prospective study, and again, many of these, they're hypothesis-free. Some of these are hypothesis-driven. So if it's hypothesis-driven, we're like, okay, we are pretty sure that this specific locus correlates with this genetic illness. So what we're going to do is we're going to recruit two groups. People with that specific, with an allele, a specific allele at that locus, and people without a specific allele at that locus, right? And so now we've got two groups, and we needed to recruit everyone, right? Everybody was recruited, so yeah, you've still got sampling bias issues, but everybody's got the same sampling bias. It's not like only your control group were people that you had to recruit for the study. No, it's like your control group and your experimental group, those with the specific allele, your experimental group, those without the specific allele, your control group, they were all recruited. So they all have the same sampling bias, meaning in some ways you don't worry so much about it, right? Yeah.
The difference between retro and pro is that retro is a specific disease and prospective is a specific
Yes.
Okay.
And then so in prospective studies, what we'll do is we will survey and sample these people for sometimes decades. So we'll recruit people, and maybe we'll recruit them from the time of birth. Again, we've got one group, they have a specific allele. The other group, they do not have a specific allele at that locus, and we're going to follow them, maybe for decades. Doing routine medical exams, looking for any evidence associated with a particular genetic illness. Okay? Does that make sense? And so that's where it'd be maybe a little bit more of like a hypothesis driven, because you're like, we're pretty sure this locus correlates with this genetic illness, but oftentimes they're still hypothesis free, where you're just like, okay, we know this locus well. We don't know exactly all of the things that it correlates with. So we're just going to look for everything. As we do these routine medical exams, we're just going to do a very extensive physical every year when these people come in. And we're going to look for anything. And we're not really trying to tie this specific allele at this specific locus to this specific genetic illness. We're just trying to see all of the pleiotropic effects of this locus. What are all the phenotypic characters that this locus is related to? Does that make sense? Okay. We're okay? All right, so now we need to talk about odds ratio and relative risk. So odds ratio, we oftentimes abbreviate this as OR because odds ratio is just way too long to write, right? Yeah, it's just copy and paste. It doesn't matter how long it is. I think I have an idea that how I could get those slides back. Anyways, I'm probably not going to do it, but I think I have something that could help. So odds ratios, these will only be used in retrospective studies. Let me spell this right, retrospective. Okay, you will only use odds ratios in retrospective studies. And we'll only use relative risk, which we'll abbreviate RR. We'll only use these in prospective studies. And so if you find yourself reading one of these papers because you're using it for your research project this semester, or just because you have a personal stake in it, in that a particular genetic illness is common in your family, or somebody you know is already diagnosed with this complex genetic illness, if you find yourself reading one of these papers, you should be able to very quickly figure out whether it was a retrospective or prospective study. And then you should be able to predict whether you're going to be using odds ratio or relative risk. Okay? And so if we're dealing with a retrospective study, we're going to use odds ratio, but these two are very, very similar. They're very, very similar. So odds ratio is equal to this. Odds ratio is equal to the proportion of, that's not the right term I want. The proportion of, and this would be a really long way to write this, but I don't know if there's a better way to write this. The proportion of people with illness and allele divided by the proportion of people without illness, but with this specific allele. So remember, in retrospective studies, what we're doing, even if they are hypothesis-free, and we just recruited people with the genetic illness and recruited people without the genetic illness, we're still hunting for anything in their genome that might correlate with that genetic illness, right? And so when it gets to the point where we're going to start calculating an odds ratio, we need to now start to make this specific for a very specific genotype. Okay? And so what we'll do is we'll take what proportion of the people with that genetic illness had this specific allele at this specific locus. We'll divide that by the portion of people without the genetic illness, the people from your control group that had that specific allele. And whatever that ratio is, it's our odds ratio. And basically the way you can think about it is, it's the odds you're going to develop that genetic illness if you have that specific allele. Right? It's your odds of developing that specific genetic illness if you have that specific allele.
So it's people with genetic illness and the allele, and people without the genetic illness that also have.
But with the allele, yeah. So without the illness, but with that specific allele.
Wait, and then the first one's also with the allele.
People with the illness and the allele.
Okay.
Yep. So I couldn't think of a shorter way to write that. In the textbook, they phrased it a little bit differently, but it's even more words than this. So you're welcome. I cut some words for you. Plus, I did with like this, so you don't have to write as much. You're welcome. Okay, and again, you can look at this, it's like, okay, given that I have this specific allele, what are my odds of developing this genetic illness? If there's a relationship, your odds ratio should always be greater than 1. right? If there's a relationship between that genetic illness and that specific allele, your odds ratio is going to be greater than 1. Does that make sense? So more people with the disease are going to have that genetic illness than people without the disease will have that specific allele. Now notice, if this is 0, we've got a problem. Do you see what problem we have if this is 0? If there are 0 people without the genetic illness that have that allele, we can't divide by 0. It's undefined, right? At which point you cannot calculate an odds ratio. 1 divided by 0 is the same thing as 4,000 divided by 0. They're all undefined. Right? Does that make sense?
So, but like if we weren't thinking about it in terms of odds ratio, would that just mean that it's like 100% correlated with the genetic?
Yeah, but you're not going to have that with these complex diseases. I mean, you're going to have that with like monogenic diseases, but you're not going to have that with these complex diseases, because these complex diseases, even if you have a really large impact, which most of these are going to be small influences, right? Specific alleles that correlate with it are going to have a small effect, but when you have several of them, then that small effect becomes a large effect. Do you remember talking about that? And then you will have few large impact mutations, but even those that are large impact, you're still not going to get 0 here. It's possible that you would get 0 here. If it was, you can't calculate an odds ratio, and what you would just have to say is that your hypothesis would be this mutation causes this genetic illness. And then you do an animal study to confirm your hypothesis.
So if it's 0, then it would just be a monogenetic illness?
Most likely, those will happen in monogenetic illnesses. You're very rarely going to have that with these complex genetic illnesses. It's possible, and if you do, you can't calculate an odds ratio, and then your hypothesis should undoubtedly be that this mutation causes this genetic illness. Because there's not a single person without that genetic illness that has that mutation, and everybody with that genetic illness everybody with that mutation has the genetic illness. So then you're like, it's perfect, let's do a hypothesis-driven study, and let's get an animal model. Let's induce that mutation in a mouse and see if they develop that genetic illness, right? Let's test that hypothesis. Yeah, Mia.
I guess I'm a bit confused. I'm wondering, like, but if you're looking at like a specific allele, then like, isn't it kind of hypothesis? Is there some kind of?
Well, yeah, but this is talking about like after you've done your hypothesis-free study, and you're just because even in the midst of our hypothesis-free studies, it's not like we have no idea what's going on. We're just not testing a specific hypothesis, right? We still have an idea, and then what we'll do is we'll just say, okay, we take our whole group of people with the genetic illness, we take our whole group of people without the genetic illness, and we start comparing them genetically. And we'll start to find loci that correlate significantly with it. After that, then we'll calculate the odds ratios for each specific locus. Yeah. Yep. Yes, sir.
If your odds ratio is 0, shouldn't that make you like raise questions about sample size?
Well, your odds ratio shouldn't be 0 either, because the only way your odds ratio could be 0 would be, oh, you're talking about undefined.
Yeah.
Okay.
That should raise questions about like sampling size or sampling bias.
Sure. Well, yeah, but if you came out to 0, like you had 0, I always want to say uninfected, but that's the wrong word. There's no pathogen. If you had 0 people without the genetic illness with that specific allele, then you have to hypothesize from that that mutation causes the genetic illness. There's really no other conclusion you could draw from that. That would be your observation, and the only reasonable thing to hypothesize is that that mutation causes the genetic illness. And if that's the case, now you're like, wow, this is really weird. This seems like a monogenic condition, where the whole time we thought it was, this really complex genetic illness that had multiple loci involved. And that's why I'm saying, like, it's technically possible, but it's probably not going to happen for these complex genetic illnesses. It'll happen all the time for monogenic genetic illnesses, right? Because those have very simple inheritance patterns.
I guess I'm just like curious of when this becomes like null and void, because what happens if like the proportion of people without the illness but with the allele just haven't
developed that illness yet? Yeah, exactly. When it doesn't become null until you develop a controlled experiment to test it, right? Because after this, you're going to assume. After this, you would assume, okay, that this mutation causes this genetic illness. And then if you do a controlled experiment for that and you induce that mutation in some animal and they don't develop that genetic illness, now you've got an issue and you're like, certainly we had some kind of sampling bias. That these people, they just haven't developed the genetic illness yet, or our samples aren't big enough. These are expensive, by the way. It's not that we do hypothesis-free studies because they're less expensive and more convenient. Now, these are expensive studies that require a lot of computer time, a lot of people working on these. You know, they'll recruit undergraduates like Nathan this summer, and they'll give you stuff like this, and they're just like, hey, you know, start working through this and calculating odds ratios for us, because it's not difficult to do, but it takes time. It's labor intensive. Okay, these are expensive studies. The benefit of a hypothesis-free study is not that it's less expensive and simpler to do. The benefit of it is it allows you to see a wider vision. It doesn't make you so narrow-minded, okay, where you're focusing on a single locus. This allows you to kind of look at a much bigger percentage of the genome. But the reality is these are tough. These are hard to do. Okay? Okay. Now, relative risk. And again, relative risk is similar to our odds ratio, except relative risk we only use in prospective studies. Okay, relative risk we only use in prospective studies. And here's how we calculate it. It is the proportion of people. Wow, where did the U come from? People with a specific allele that develop, well, this is just, I don't know how to shorten this, I'm sorry. Develop the illness. Okay? The proportion of people with a specific allele that develop the illness, whatever that genetic illness is, divided by the proportion of people with a specific allele that never develop, develop the illness. And again, in many ways, odds ratio and relative risk, they're very similar. You're basically, you're getting to the same point. Over here, you're like, okay, what's your probability of developing this illness if you have this specific allele? You come over here, and you're like, what's your probability of developing this genetic illness if you have this specific allele? You get to the same point, but the way you get there is a little bit different. Because in a prospective study, you started with one group has a specific allele, or has a specific genotype. Another group, they don't have that specific genotype. And now you're studying them to see if and when they develop the genetic illness. But again, of course, we're going to have issues here. Because what if this person would have developed it, but they died before they got to an age where they were going to develop that genetic illness? Or they would have developed the genetic illness, but something in their environment just prevented them from developing that. Right? You get some kind of phenocopy, right? Where you've got the genotype that is almost always going to mean you develop that genetic illness, but it never happens, right? Because, I mean, you've got a wonderful environment that just keeps you from experiencing that. Bless you. But all of this is basically to get some kind of a ratio to tell us how much more likely are you to develop the genetic illness given that you have a specific mutation. And whether you're talking about odds ratio or relative risk, they both come down to the same thing. It's like, how much more likely are you to develop that genetic illness given that you have this specific mutation? Okay? We just come about it different ways. There we come about it from a retrospective study. Here we come about it from a prospective study. Jack.
Well, the only difference is with odds ratio, you start with the disease and look at the allele.
Yes.
With relative risk, you start with the allele.
Yes.
And the disease.
And look at the disease. Now you can do a prospective study where you have two groups and you basically randomly assign people to there, and you're not looking at their genotype at all at the beginning. You just randomly assign people to two groups. Okay? You randomly assign people to two groups, and you can even do prospective studies where you do something. Right? So like you randomly assign people to two groups, and in one group, you give them an environment that you know actually can prevent the development of a particular genetic illness, right? So like you encourage them to have this specific diet, to do these specific things, but I mean, these are decades long. Like, you can't control what people do, right? I can't even control what I do, let alone trying to control people. And it's like, hey, for the next 5 decades, I need you to eat a diet that's very low in red meat. No thanks, doctor. I don't care. I'll take the genetic illness and my red meat. Yeah.
So is that one like living people and this one's like dead? Like
No. They're both living people. It's just this one. It's like you can execute a retrospective study in a short period of time, right? The bulk of the time spent in a retrospective study is getting participants. And so getting people with a genetic illness is very easy. Most of these studies are done by physicians, right? Done by physicians that also do research. And it's like you've got a physician that is a a nephrologist, right? You've got a nephrologist, and they're wanting to do some research on a specific kidney disease, right? And they've got tons of patients coming to them all the time to recruit for these kinds of retrospective studies. And then all they've got to do is recruit people without that genetic illness. The beauty is, you've got family members of the people that have that genetic illness, right? And if they don't have any pathology, you're like, hey, you want to be part of our trial? And it's like, hey, it might help your brother or your dad. Like, sure, I guess. And then they're genetically similar too, and it'd just be a wonderful situation. But then here, yeah, so the bulk of your time there is basically spent recruiting participants. Because once you have your participants, then you just, okay, we take a saliva sample from everybody, we sequence very specific genetic markers, and then we just compare the two groups. And we're done. Like, your involvement in that trial literally took 35 seconds for you to fill up a vial with saliva. And you're like, well, yeah, but if you can't produce a lot of saliva, it might have taken a minute instead of 35 seconds. These are long-term studies. These tend to be done like when, these happen a lot when there's a specific genetic illness that runs in a family, where it's not normal Mendelian inheritance, but it's a lot more common in that family than it is in any other family. So they'll do a prospective study where they'll follow people from a specific family, and then they'll just recruit other random people, and they'll follow them for decades. Like, there have been studies, a lot of studies in the UK following very specific families. And I'm forgetting what some of the studies, we talked a lot about it last year in genetics. I'll find some of these papers and I'll send them to you. But there's like a 3 or 4 decades long genetics research project that's been going on, studying basically all of the members of one family and then like the rest of the UK. And they're looking at it as like, okay, what's the probability of this family developing this genetic illness, or a random person from this family, compared to just a random person from the entire UK. But ultimately, you get to the same thing. It's just these are done a little bit differently. They're done, I mean, one thing is like, if you want to submit a grant for this kind of project, that's wonderful. Because you're like, I'm going to submit a grant that's going to pay me $100,000 for the next 40 years to carry this out. I mean, that's pretty decent job security, right? Whereas here, you submit a grant and it's like, man, I need $100,000 to recruit people, and the study's going to be done in 5 seconds, and then it's over, and then you publish it, and this is going to get you 1 publication. Doing something like this might get you 50 publications over your career. Like, every year you generate another publication where we understand more about the relationship between these specific mutations and this genetic illness. And it's cool stuff. Does that make sense? Okay. That's odds ratio and relative risk. I think I did a better job of explaining it now than in the initial lecture that I recorded for you. And I did have a nice slick slide for it, but that slide's gone. And that one never got onto the recording. Actually, you know what, it did a little bit, because it was the next slide to come, and it's like a little version to show you. But it's probably just the first point on that. Yeah, it's just gone. I mean, it's just gone. So, any questions? Yeah.
I just want to make sure that
Yeah.
let's say like with a specific allele,
Mhm.
is it not?
This is, yeah, it's a specific mutation. Both of these get to the same thing. Like, given that you have a specific mutation, what's your probability of developing the genetic illness? And both of them get to the same thing, they just go about it different ways. Okay, there aren't any questions. We'll go to the slides that I put together for today. And so you have seen these key terms, and we'll go through these in a little bit more detail. But before we do that, are there any questions on any of these key terms? While I was recording the video, I got a little messed up by purifying selection. And I don't know if you noticed that pause. I was like, wait a minute, because typically when you learn natural selection, you learn directional, stabilizing, and disruptive. And I don't know if you remember all that. I'll show you that in a minute. But then I got a little weirded out, and I was expecting disruptive to come next, and it didn't. Purifying came next, and then I don't know. I freaked out. And that's why the video ended up being 10 minutes instead of 9, is because of my emotional distress. I lost myself. So, any questions on these key terms? Nothing? All right, cool. This makes it simple. All right, now typically when you learn the mechanism, or you learn the flavors of natural selection, okay, you learn directional, stabilizing, and purifying. And here's what you would do. You'd set yourself a nice little axis, and here on the x-axis you would do phenotype. And then here on the y-axis, you would do frequency. And you really only do these, you really only talk about directional, stabilizing, and disruptive, which wasn't mentioned in this chapter and where I was going with this, when you're dealing with QT characters. Do you remember this term? QT characters? Quantitative traits. Do you remember that now? Quantitative traits? Maybe? Okay. Quantitative traits are where you have basically a minimum value or a minimum version of that phenotype. You have a maximum version of that phenotype, and everything in between is possible.
Right, like height.
Like height, muscle mass, intelligence. What are some other things?
Like intelligence hard, there's not like an axis, there's like so many other variables.
Yeah, but there are ways we can quantify intelligence. You could challenge the quantification of intelligence, but there are ways we can quantify intelligence. Like if you do IQ you can plot as a phenotype, and it would be a QT, it would be a quantitative trait. Basically, I don't even know what the lowest IQ ever recorded is. I don't know. The highest IQ ever recorded by most metrics is like 165, 170. But then some metrics, all of a sudden you have somebody score 200, and you're like, what does that even mean? That's ridiculous. But anyways, sorry. Maybe I'm just feeling self-conscious because 200 is way larger than my IQ. I don't know. Okay, so we construct something like this, and again, it's only for quantitative traits. And so basically we've got something that looks like this with most of these quantitative traits. Man, that is a beautiful bell. Wow. And so we end up with some kind of an average phenotype. Okay? And so let's do human height as an example. Okay, so human height, you'd expect to see something like this. If you plotted the frequency, that is the proportion of the population with any particular height, and you plotted that against height, you'd end up with something that would looks like this. It'd be a little bit wider, because the minimum human height's really, really short, and the maximum is really tall. But you'd end up with something that looks like this. Now, in directional, so directional selection, what that's going to do is that's basically going to move. Wow, I did it again. Wow, so directional selection is basically just going to move everything in one of the directions. It's going to move your average trait to the right in this case. So let's say if we're talking about height, the average height is going to increase, and basically everything's going to move in that direction. Your entire bell moves in that direction, yeah.
What causes it to move? Like,
Well, it depends on what it is. For human height, there's nothing really that has done this. I mean, better nutrition, but that's not really, you wouldn't see this anyways. But where you see this a lot happens, I mean, you can look at it in the past with certain human characters, but you see it happen a lot, like our textbook example of natural selection are the Galapagos finches, right? So the Galapagos finches, what is it, Peter and Rosemary Grant? I think those are their names. Anyways, it's a husband-wife team, and they've been studying Galapagos finches for like 5 decades, and have published hundreds of articles. And you could see this kind of a relationship with beak size, where the average beak size will increase based on environmental conditions, and then it'll decrease, and it kind of just fluctuates over time. But with like a human character that would have done this, where we could actually see strong evidence of directional selection, might be like pigment type, or not pigment type, but pigment concentration, where you could see it move in this direction. A drop in pigment concentration as our species started to move further away from the equator.
so the actual data, like in the graph, does not change, just like the values of the data.
Right, the average value is changing and is shifting in one direction under directional selection. Okay, now stabilizing, I'm going to draw this one in the middle. Stabilizing selection, you get something like this. That one doesn't look as good. This still looks okay though. So stabilizing selection, your average isn't changing, and you're getting fewer individuals at the extreme. Fewer individuals at the extreme. Okay? So directional, your average is changing, and basically everything's moving in a particular direction. Stabilizing selection, your average isn't changing, you're getting fewer individuals at the extreme, and then typically what would come next. Oh, perfect. Got another color here. What would come next is disruptive, and I'll draw this down here, and then I'll draw it on the graph. Disruptive selection, and there's where you would have something like this. Where you actually start to have fewer people with that average condition, but your average really doesn't change all that much. You just start to make the extremes more common. And so this, for human height, you're like, what on earth would lead to that? And I mean, the only thing that would potentially lead to that is if the average height, for some reason, you were less likely to survive if you were of average height, which I don't know what that would be, or sexual selection, which is a type of natural selection, right? Where tall people tend to mate with tall people and short people tend to mate with short people, and you end up with fewer people that are average. Yeah. But notice, we didn't talk about this, but this is what I was anticipating coming next. But purifying selection, you would never graph it like this. Because what purifying selection means is you're just, natural selecting is selecting against every single allele other than one. Natural selection is selecting against every single allele other than one. Only one allele for a particular gene is persisting from one generation to the next. And so purifying selection tends to happen in our housekeeping genes, right? In the genes that those genes, their expression is mandatory for the survival of that organism. And there's nothing you can do. Any mutant is going to be bad. Any mutant in that is going to be bad. A lot of the enzymes that work in key metabolic processes, like glycolysis, or one of the machines that work in oxidative phosphorylation. Okay, these are going to be subject to purifying selection, where any mutation is going to be selected against. Okay, and it's not going to be a quantitative trait. It's going to be more like a single locus situation. And that's why I wasn't really ready for it, even though I wrote these key terms out. I should have been ready for it, but I wasn't. I can blame tons of people but I'll take it. It's all me. It's all me. Okay. Now, balancing selection. So these two terms, these are dealing with quantitative traits. Okay? These two are types of selection that you see in quantitative traits. These two are what you see in discrete traits, where you basically, it's an either or. It's an either or. Again, these are discrete traits. It's an either or. You have this version of the protein or you have this one. Single loci stuff. Single locus, sorry. Subject, wait, that would be adjective noun agreement. Okay, a single locus. So purifying selection, you select against every allele except one. Balancing selection, two alleles, at least two, are being maintained. At least two alleles are being maintained. And being maintained at approximately the same frequency. Being maintained at approximately the same frequency. And the main reason for that, and this is why I put this term with this, the main reason why you have balancing selections where you maintain at least two alleles at about the same frequency is because there's some advantage to being a heterozygous individual. There's some advantage to being a heterozygous individual. Now, there's one really good example of this in humans. And what is it? Sickle cell trait. Okay? Now, we're just beginning to understand the mechanism behind how people with one mutant copy and one normal copy of that gene, you could call it hemoglobin S. It's in the beta protein. But the gene I think they call hemoglobin S, and then the actual protein product is the beta chain of the anyways, sorry. It's undoubted that having one mutant copy and one normal copy makes you significantly less likely to die from malaria. It even makes you significantly less likely to ever experience any pathology associated with malaria. Even a small, minor reaction. And so what that has done in areas where malaria is endemic, it's maintained two alleles. The mutant allele, that if you have two copies of it, it'll cause sickle cell anemia, and the normal allele. Okay, it's maintained it at approximately the same frequency for several human generations. Now, in the textbook, it also talks about some ideas that people have had for why certain genetic illnesses that develop before you reach sexual maturity and can actually reduce your fitness to zero, why they persist. So a good example of this is cystic fibrosis. So cystic fibrosis tends to, you start to experience pathology associated with it, even in utero. In utero, you are already experiencing pathology associated with having a mutation in that CFTR gene. By the time you reach 5 or 6 years old, you're typically starting to experience some very intense pathology. Dealing with lung infections regularly, dealing with the inability of your kidneys to efficiently get rid of metabolic waste. And in males with cystic fibrosis, you have almost a 100% sterility rate, which means your fitness is reduced to zero. Right? If you are sterile, your fitness is reduced to zero. And so the question is, how could this possibly persist? Well, one, the heterozygous individuals don't experience any pathology, right? So that's already going to help you. If you're heterozygous, you have one mutant copy of that gene and one normal. That one normal copy is enough. There's no heterozygous insufficiency, right? Where if you only have one copy, you're not going to make enough of that membrane protein. No, one is enough. So that's part of it. But then the other thing is you've had some teams that have proposed that being a heterozygous individual there actually prevents your development of certain pathologies. And the textbook talks through that. The reality is there are no data to support that hypothesis. It's kind of just an idea behind why do certain genetic illnesses still exist. But the reality is we don't need to explain this because natural selection has a very small impact on humans, right? We've talked about this before. We'll talk about this time and time again. Natural selection is a very weak force when it comes to humans. Why? Because we do whatever we want. We change our environment, right? We don't really allow our environment to have a huge impact on our survival. And certainly not on our reproductive success, right? So, and the reality of that is, why can't natural selection rid these monogenic genetic illnesses that even reduce fitness to zero? And the thing is because we don't care. I mean, obviously, if your fitness is reduced to zero, there's nothing you can do. But your siblings that oftentimes are going to be carriers, right? You got 2 mutant copies of that CFTR gene. Your siblings without cystic fibrosis, they're almost certainly carriers. They have to be. Because if you got 2 mutant copies, both of your parents had to be carriers, which means all of your siblings that don't have cystic fibrosis are carriers. And guess what? They're passing that mutation off to the next generation. I mean, it's just like, we don't allow natural selection to really influence very much about human evolution. So most of the things, when you can find evidence of a change in the human genome, it is very rarely going to be the result of natural selection. There are some good examples, and one I gave you was the amount of melanin production. That's one that you can see as humans spread around the globe and started to get further away from the equator into places where sunlight is much less direct, and where sunlight is a lot less predictable. You can see that the amount of melanin production has decreased. I mean, you can see it now. You can see it now as people move from an area, where their ancestors lived, where they had a lot of melanin production, so near the equator, Africa, Asia, South America, to other places. You can see that every generation, the amount of melanin they produce decreases. Okay, so that's one. But other than that, most often when you find evidence of an actual change in the human genome, and by the way, you might be thinking, how would we find evidence of this? It's really wonderful. We find old humans that are buried, and we isolate DNA from them. And then we compare those genomes to modern human genomes. Right? And it's like, oh, this is so cool. It's like we've got a snapshot. Somebody just saved this genome for us, ready for us to use. And you're like, wow, it's really weird that DNA could survive for that long. Maybe this person that's buried isn't really as old as we think they are. But anyways, that's another conversation for another time. So most often, the change in the human genome is the result of horizontal gene transfer or genetic drift. Almost always. That we can tie the actual change in the human genome to horizontal gene transfer or to genetic drift. And you're like, horizontal gene transfer? I mean, we're not bacteria, right? We're not undergoing conjugation where like one human would come stand next to you. It's like, listen, I feel bad. I feel bad for all of you that aren't redheaded. So I'm going to give you the ability to deposit red pigment into your hair. I mean, really, I would love to do that, because I do actually feel bad for everyone who isn't redheaded. I don't know why. Maybe it's because redheadedness is disappearing, so I feel unique, And I want you all to feel unique as well. But it's not like, so bacteria could just be like, hey, I'm going to give you the redheaded gene, I make a little pillis and transfer the genetic material to you. Like, that's not how it works in humans. But we still have one of our 3 main mechanisms of horizontal gene transfer in bacteria happens all the time in humans. And it's transduction. The movement of genetic material from my genome to yours by a virus. And you're like, this is so cool. That is so cool. Now, of course, if we just allow viruses to do what viruses are going to do, it's a little bit random and unpredictable. But if we design viruses to actually transfer a very specific... Are you going to say, have we done this?
Yeah, you said this is one of the most common ones.
No. No, this is the only one that's active in humans. This is the only mechanism of horizontal gene transfer that's active in humans. And currently it's illegal in almost every country, and it's internationally illegal to design viruses to genetically modify humans. But that doesn't mean it doesn't happen, right? I'm just saying it's currently illegal. So is driving over the speed limit. you know? Oh, sorry. No, this is no joke. I'm not trying to make a joke. Genetically modifying humans is not a good idea. But what I'm saying is we find enormous evidence of it happening in the history of our species. Enormous evidence of it happening in the history of our species. And it's so cool because you can take one people group and you can compare their genome to another people group, and you're like, man, this people group, they have this gene that you don't even find in this other people group. You're like, how did it get there? And you're like, well, either the wild type state was it was there and some people groups have lost it, or the wild type state is it was absent and some people have gained it. And the only way you could have gained it is for a virus to give it to you. And what we're starting to do is we're starting to find a lot of evidence of not just horizontal gene transfer from human to human through human viruses, but horizontal gene transfer from bacteria to human through bacterial viruses. Through viruses that infect the bacteria in your gut, but also infect your cells. Especially if those bacteria are intracellular bacteria. And the viruses, in order to escape and find more bacteria, they have to leave your cell. But sometimes they don't. Sometimes instead they go lysogenic and put their genome into your cell. And it's just so fascinating. And for a long time, we knew this has nothing to do with humans. Well, not nothing. This is very little to do with humans. For a long time, we knew that the gene that black widow spiders use to make their venom is found in other insects. And the assumption was that maybe this was like a gene for an immune response, right? It was a gene for an immune response, and over evolutionary time, some lineages of arthropods learned how to use it as a weapon, right? And so learned how to actually make a toxin out of it. And you're like, that's so cool, it's such a wonderful story. But then people were like, let's actually go and sequence the gene in these insects that aren't black widow spiders. And you know what they found? The gene was surrounded by obviously viral DNA inside of the genome of the insects. So now it's not like this wasn't a gene that existed as an insect immune response that then was weaponized in some lineages. This was transferred to some insects by a virus. And you're like, that is so cool. And you're like, okay, so there are bacteria that infect almost all arthropods. And one of those genera is called Wolbachia. Okay? Wolbachia, that's the genus. This is bonus information, but it's so cool, I have to share it with you. So this genus of bacteria is called Wolbachia, and it affects almost every arthropod species. And it's got a phage, a specific virus that we know well, and we call it the Woe phage, because it infects Wolbachia. And you know what they found inside of Woe phage when people started looking for it? They looked inside of a Woe phage that they pulled out of just some random insect, and you know what was in that Woe phage's genome? The gene for making black widow toxin. Just in some random Woe phage pulled from a random insect, the gene for making, it's called latrotoxin, in case you're curious. And you're like, how much of this is happening? This paper was just published 2 years ago, by the way, maybe not even 2 years ago, and it's basically transformed our entire idea about genome evolution. Because it's like, there's no doubt that there are mechanisms in place to facilitate horizontal gene transfer, and a lot of it, among domains even. And now we need to start sequencing viral genomes and finding how much of this is happening. And then it's like, man, why do viruses exist? It's another story for another time. Okay,